Male reproductive anatomy and spermatogenesis. To begin, we're going to take a survey of the male reproductive organs. The most significant of these would be the testes. This is the organ that creates sperm. They are housed in a pouch of skin and smooth muscle called the scrotum. And the scrotum's function is to regulate the temperature of the testes. The male testes are, of course, held outside of the body in an effort to keep them a little bit cooler than body temperature for optimal sperm production. Now, inside of the testes, there is a bunch of coiled tubes called the seminiferous tubules. So a network of coiled tubes, this is the location in which sperm are reproduced. Then, those sperm that get created in the seminiferous tubules are sent to a structure on top of the testes called the epididymis. The epididymis is a coiled duct on the surface of the testes where the sperm are stored and allowed to mature further. Taking a look at the picture here, we can see a sagittal section of the male, that's picture A, and we can also see an anterior view. In this picture, we've got the testes, and if you take a look at the picture in B, you can see that they've kind of taken a section of the testes, and there's a bunch of coiled little tubes inside called seminiferous tubules. They're going to then connect to a structure on top of the testes called the epididymis. The epididymis then allows sperm to move into something called the ductus deferens, or you may have heard of it called the vas deferens. This is the tube connecting the testes to a structure called the ejaculatory duct. The ejaculatory duct then is going to be located inside of the prostate gland. It connects with the seminal vesicle, and it allows both the vas deferens and the seminal vesicle contents to be released into the male urethra. The penis is the copulatory organ of the male. This is the organ that's used to insert sperm into the female reproductive tract. It delivers sperm, like I had mentioned, into the female reproductive tract. And it is responsible for maintaining an erection in order to be a copulatory organ. Erection, by definition, is an increase in length, diameter, and stiffness, and we'll look at the mechanism behind this in a little while. Mostly, it's due to an increased blood flow into the penis and decreased blood flow out of the penis. Taking a look here, we can see the vas deferens coming off the epididymis of the testes. It travels upwards over the male urinary bladder and into the ejaculatory duct that you can see inside of the prostate gland. Additionally, the seminal vesicle empties its contents into the ejaculatory duct. There are some accessory glands that are going to release their contents into semen. However, sperm don't necessarily travel through these structures. So, the first of those is the seminal vesicle. These are located on the posterior side of the urinary bladder. They are responsible for releasing fructose for the sperm. These sperm are going to have lots and lots of mitochondria associated with them, and we know from that they will be able to undergo cellular respiration, so they're going to need a fuel source. The fructose acts as a fuel source for those sperm. It also releases something called prostaglandins. Prostaglandins are local hormones. They are going to produce smooth muscle contraction in the female reproductive tract in order to pull the sperm up closer towards the cervix. The prostate gland is another of the accessory male sex organs. This is located underneath the urinary bladder, and it completely surrounds the urethra. So the urethra is going to run right through the center of the prostate, which is why you see all the commercials on TV for an enlarging prostate, having a man maybe need to go to the bathroom more frequently, or even inhibit that flow of urine. The prostate releases an alkaline fluid that's used to buffer the vaginal environment. So when the sperm gets sent off, that the low pH inside the vagina doesn't kill the sperm before they have an opportunity to come in contact with an egg. Taking a look at this picture again, we can see on both the sagittal section and the anterior view, our seminal vesicles, as well as the prostate gland that's located directly below the urinary bladder. The other of the accessory glands is something called the bulbourethral glands. These are located at the base of the penis. They release a mucus secretion, and its function is to help lubricate the glans penis. The glans penis is what you probably commonly call the head of the penis, and it also helps to wash away any acidic urine that's left inside of the male urethra. So again, taking a look here, if you look for the prostate gland, right below it are paired bulbourethral glands, and they're quite small. We're now going to look at something called spermatogenesis. Spermatogenesis is the process by which we create sperm. This occurs, again, in the seminiferous tubules of the testes. And we begin with a cell called a spermatogonia. This is an undifferentiated cell with the normal 46 number of chromosomes. The spermatogonia then are going to differentiate and become primary spermatocytes. They're going to divide by mitosis, which is simply increasing the number of cells. We have not done any chromosome division yet, or decrease in chromosome number. These cells, again, contain that 46 or normal number of chromosomes. A secondary spermatocyte is then when we undergo meiosis. Meiosis is the process by which we decrease our chromosome numbers by half. The cells now have 23 chromosomes. And really that's what we want, because when mom and dad give their gametes, so an egg and a sperm, to the new zygote, we want to have a number of 46 restored. But we have to have half the information from mom and half the information from dad. Taking a look at this picture, we can see the spermatogonia inside the cross-section of the seminiferous tubule. They're the ones to the left. Then we create primary spermatocytes. They undergo meiosis, and we're going to create a secondary spermatocyte. All the time you can see that these cells are moving closer and closer to what would be the lumen of the seminiferous tubule, or the center where the sperm can start to mobilize. Now, the next differentiation that we're going to have is something called spermatids. These contain 23 chromosomes. They're going to mature into a mature sperm. And the cells that make up the walls of these seminiferous tubules are called Sertoli cells. These are supportive cells, and they are simply going to help regulate the number of sperm created and make sure that the nutrient and such that these sperm need are in place. So as you take a look at this picture again, you can now see the spermatids. They're starting to look more and more like a mature sperm that's going to get into the lumen of the seminiferous tubule. And additionally, you can see the light purple dots in which the sperm cells are reproducing and differentiating. Those are your Sertoli cells. So in this picture, you can see about 2 Sertoli cells. Now, the sperm itself has a couple structures. One is a head, and on the tip of the head there's a structure called the acrosome. The acrosome contains digestive enzymes, and it's used in order to penetrate the egg. The mid piece is going to have mostly mitochondria in it. That's going to be used as an energy source so that the flagellum can propel. And the tail then also acts as a propelling structure. It is mostly flagella.